All right, welcome back, everybody. We're going to continue with our content as far as macromolecules is concerned. So let's go down to where we were. All right, so yesterday we were talking all about, or yesterday, last video, we were talking about fats, lipids. As far as lipids are concerned, we talked about saturated versus unsaturated. We also discussed cholesterol as far as HDL and LDL. So make sure we're good with that. Today, we're going to be focusing a little bit more on nucleic acids. Now, before we get into nucleic acids, I do think it's worthwhile talking a little bit of history. Obviously, this isn't a history course or anything like that or a history video, but it's worthwhile understanding the science behind it and some of the injustices that occurred with regards to this. So we've known about DNA for quite some time, but not until the 1950s were we able to actually observe what the DNA double helix looks like, discovering the DNA model, as it's called. Watson and Crick, an American and English scientists, they were credited with discovering the model in 1953, which doesn't seem that far back, or seems kind of ancient since a lot of us weren't born in the 50s. But if you think to how long humanity has existed, it is very, very recent. If we're thinking in those terms, here's the issue though. Watson and Crick were not the only two working on discovering the DNA helix. They had a third, Rosalind Franklin. Rosalind Franklin, her work was essential for this. It's been Her work was studied and analyzed, and it's been shown that without her work, Watson and Crick would have likely never found the DNA model, or maybe even took them a couple more years to discover it, or maybe even someone else would have discovered it. So she was essential. But unfortunately, in this time period, especially with the way that women were treated and treated unequally, um, she was never credited. She was never credited for the discovery. Her work was, again, necessary for this, but many times people would approach them in their laboratory and in the scientific setting and believe she was simply a secretary. So this is something that eventually was addressed, eventually was changed, eventually edited in textbooks. But it is just one of the many numerous instances of women, minorities, people of color who are discredited for their hard work and their legitimate work too. She was an excellent scientist. So it's one of those minor things I like to bring up, that we should be continuing to push the envelope on this. It still happens to this day, not as often as back in the 50s, obviously, but we still have to make sure that the people that deserve credit get their credit. Okay, let's talk about nucleic acids. Nucleic acids are a group of macromolecules, right? It's the third macromolecule we're talking about so far. The unique thing about nucleic acids is that they're not consumed, as in we don't get nucleic acids from what we eat. Nucleic acids, we synthesize them, we make them. We make them from the stuff that we typically break down, right? So first and foremost, I do want us to understand these atoms that are associated to nucleic acids. It's stuff that we talked about in the very first couple slides. So make sure you're familiar. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Those elements are essential for nucleic acids to be formed, especially phosphorus. It's unique because phosphorus is only found in this macromolecule compared to the other four. The unit of a nucleic acid is a nucleotide. Nucleotide. Nucleotides are made up of three major components: a sugar, which we talked a lot about with regards to carbs; a phosphate. This is pure chemistry, but a phosphate is a phosphorus with four oxygen molecules. Don't have to worry too much about that for anatomy. And bases. Bases is really big with regards to how nucleotides work. A base is going to be these chemical puzzle pieces. What are the bases we need to know? Cytosine, guanine, this is a slight typo here, adenine, Adenine, thymine, and uracil. So those are the five bases we need to know in regards to nucleic acid. There are two types of nucleic acid, DNA and RNA. Everyone's heard of DNA before, right? At least the basics of DNA. RNA doesn't normally get as much love. I like to joke that it's like the redheaded stepchild in this situation. But RNA plays a very important job, especially when it comes down to protein production. So the function of nucleic acids, as you can see here, depends on which nucleic acid are we talking about. For DNA, it's all about genetic material. It is literally our genetic code. Whereas RNA is going to be all about creation of protein, which is essential. Protein is not just, oh, I want to consume protein so my muscles get big. Far from it. The regeneration of tissue in the body relies heavily on protein use. So, and not to mention, many proteins make up more complex structures like enzymes, which we do talk about in this unit. So, proteins are super, super important. We got to make sure that we're sustaining ourselves amply. Okay, let's talk more about DNA. First off, I would know what DNA stands for: deoxyribonucleic acid, as far as the three letters. Deoxyribo, that tells you what sugar. As you can see here, the sugar found in DNA is deoxyribose. So I would know that sugar. I would make sure we're comfortable with that. I'll try to underline some stuff while we can, to the best of my knowledge, or to the best of my ability, not using a pen. Um, Nucleic is simply telling us that it's a nucleotide, and then it's an acid as far as a chemical structure is concerned. DNA is the hereditary material passed from parents to offspring. We all exist because our mom and our dad blended genetic material together, and more specifically, underwent what's called genetic shuffling. Genetic shuffling allows for our species to survive. If every single one of us were identical, and the only differences were some of us were men and some of us were women, so we could procreate, our species would be long gone. Because the problem is, there's such a thing called mutations. And I'm not talking about X-Men level mutations, right? We're talking about mutations that can lead to babies dying before they're even born. We're talking about mutations that can lead to people dying before the age of 25. These are serious level stuff. And mutations have a much higher risk when you're mating the same genetic material over and over and over again. It's a big reason why the royal families, especially in the medieval era, when the royal families ruled everything as far as the major empires in that time period, they polluted their bloodline. They were under the belief that this pure blood was necessary to maintain. So they would purposefully procreate with their sisters and brothers and sometimes cousins, but obviously most of the time it was with the immediate family. And when that happened over generations, that would cause dramatic mutations and dramatic changes to the individuals that were being born. Now, as far as the structure of DNA is concerned, we know it's a double helix, right? Watson, Crick, and Franklin, they talked about it. It's this typical model. The sugar is a deoxyribose. That's what I talked about earlier. Now, the bases, if you look at the bases in this picture, you'll notice that it's almost like puzzle pieces matching each other. G matches to C, A matches to T. As long as you can remember that those four bases are the ones found in DNA, we're good. So cytosine always pairs to guanine, adenine always pairs to thymine. My mnemonic, I love mnemonic devices. I think mnemonics really work. It's like a shortcut for your brain. I learned this mnemonic in biology, high school in year 2005. So it was a long time ago. All tigers can growl. A, T, C, G. A pairs to T, C pairs to G. If that mnemonic doesn't really work for you, then it's fine. No problem. No worries. But I think they help. All right. What's next? We got RNA. RNA is, again, related to DNA. In fact, to make RNA, you need DNA. This whole transcription process that you should remember from biology. But RNA is going to be the specialized strand. It's unique, it's similar but different. It's really important for a concept called translation. This is stuff that you typically learn in biology. Translation is when you decode RNA and turn it into protein. So here's some key distinctions, key differences that you do need to know that you should see coming up in my class. First of all, RNA is single-stranded. Compared to DNA, where it has two strands, RNA is just one single strand. Secondly, when you look at the nucleotide, some of it is similar, right? There's a phosphate group, it's just like in DNA. The sugar is also there, but the sugar found in RNA is a different sugar. It's called ribose, whereas DNA had deoxyribose. You don't need to worry about the chemistry as to what is a deoxyribose and what's a ribose. Long story short, deoxyribose has a couple extra oxygen molecules, and that's what makes it a unique sugar. And finally, the bases. Cytosine, once again, pairs with guanine. We saw that with DNA. But adenine is going to pair up with a new base. Adenine is actually going to pair up with uracil. Uracil replaces thymine in the instance of RNA. Make sure you know that. I will 100% ask questions associated to this as far as which of the following are differences between RNA and DNA. So as long as you can identify RNA is single-stranded, has a different sugar, and has a different base instead of thymine and replaces it with uracil, you'll be good. No worries there. Let's erase. All right. Next, this is pure chemistry, right? You do need to obviously know what all of these are: adenine, guanine, cytosine, thymine, and uracil. But what I do find interesting is that when you look at the chemical structure of thymine and uracil, they're almost identical. The only difference is this CH3 structure. Again, a little bit of chemistry for you guys, but that CH3 structure is known as methyl. When you add a methyl to a uracil, voila, you now have a thymine. They're very similar, but obviously different bases. Okay, lastly, for today's video, purines versus pyrimidines. As far as purines and pyrimidines are concerned, our bases, we can summarize them or categorize them, should I say, based off of their structure, based off their chemical structure. Purines have 2 rings, whereas pyrimidines have 1 ring. So the mnemonics I like to use, first of all, the mnemonic for pyrimidine I think is a lot easier. The pyrimidine mnemonic I think of is pyramids cut. Pyramids cut. Because look, thymine, cytosine, and uracil are considered pyrimidines. So what I think of is, okay, well, pyramid, pyrimidine, cut, C-U-T, cytosine, uracil, thymine. That works for me. The other mnemonic for purines, I do think you have to have a chemistry background. If you've taken chemistry before, this other mnemonic could be really helpful for you. Pure as silver. Pure for purine. Silver in the periodic table of elements is Ag. So A is adenine, G is guanine. That works for me too. But again, if any of these mnemonics are a little confusing and you're like, wait, what? What does that mean? I can 100% help you out there and figure it out with you and just say, don't worry about it. Memorize it if you need to. You're good to go. Okay? So that's where we're going to finish. Great place to end. Like I said, pretty short unit. We're going to transition from our discussion on nucleic acids straight into protein. So that'll be our next stuff that we're going to go over. All right, thanks, guys. Thanks for tuning in. Hope you have a great rest of your day. See you later.